Hello everyone, welcome to my channel Microbiology with Shabu. In this video we are discussing about the history of microbiology. This is the second part. I have already uploaded the first part in my channel. You can watch the first part by clicking that icon on the right top. So let's start. Development of pure culture technique. Pure culture means a culture containing a single species of cells. The method of obtaining bacteria in pure culture using salt media was first developed by Robert Koch. At first he cultured the bacteria on the sterile surface of cut boiled potato. This was unsatisfactory because bacteria would not always grow well on potato. He then tried on semi-salt liquid medium by adding gelatin. Separate bacterial colonies were developed after the surface have been streaked with the bacterial sample. But gelatin was not an ideal saltifying agent because it was digested by many bacteria and liquefied at 25 degrees Celsius. A better alternative was proposed by Fanny Elskimius Hesse, the wife of Walter Hesse, one of Koch's assistants. She suggested the use of agar as a saltifying agent. She had been using it successfully to make jellies. Agar was not attacked by most bacteria, saltifying at 40 degrees Celsius and melt at a temperature of 100 degrees Celsius. One of Koch's assistants, Richard Julius Petri, developed the Petri dish for solid culture medium. This development made a possible isolation of pure culture that contained only one type of bacteria. Louis Pasteur and his major contributions. The development of microbiology as a scientific discipline dates from Louis Pasteur. Though trained as a chemist, his studies on fermentation led him to take an interest in microorganisms. He established that fermentation was the result of microbial activity and the different types of fermentations were associated with the different types of microorganisms. He discovered that carefully selected yeast make a good wine, but the mixture of other microorganisms made a taste sour. He suggested that proper selection of microbes yeast could ensure consistently good product. To achieve this, he destroyed the microbes already present in the grape juice by first heating it. After cooling, inoculated the juice with selected yeast cells. He also observed that finished product could be preserved without any change of flavor if it was heated to 56 degrees Celsius for 30 minutes. This technique was later on known as pasteurization. Louis Pasteur was requested to study about the silkworm disease which was threatening the silk industry of France. He identified their causative agent as a protozoa Nosema bombiasis. He suggested to select healthy, disease-free silkworm to avoid the disease. By this time, in 1859, he has done his famous Swanneck experiment to disprove abiogenesis. In 1880, Louis Pasteur isolated the bacteria responsible for chicken cholera. While experimenting with these bacteria, he noticed that the bacteria loses its ability to cause the disease after it was grown in laboratory for long periods. However, this virulent strain was able to induce immunity against subsequent infection by its virulent strain. He also observed that Bacillus anthracis, causative agent of anthrax, lost their virulence on cultivation at a high temperature of about 42 degrees Celsius. But the bacteria were highly pathogenic at their usual growth temperature of 37 degrees Celsius. He called this phenomenon as attenuation, and aged culture of chicken cholera became the first attenuated strain. Later, Pasteur called the attenuated culture a vaccine in honor of Edward Jenner because many years earlier, Jenner had used vaccination with material from cowpox lesions to protect people against smallpox. Pasteur then developed the rabies vaccine. He produced the disease in rabbits by inoculating them with saliva from rabid dogs. When an infected rabbit died, the brain and spinal cord were removed and dried. He tested this preparation in animals and found that they gain immunity to rabies infection. In 1885, he tested this vaccine preparation in humans. A boy named Joseph Mister was bitten by a rabid dog and his family compelled Pasteur to inoculate the child. This was a great success and the boy was saved. In gratitude for Pasture development of vaccine, people from around the world contributed to the construction of Pasture Institution in Paris, France. One of the initial tasks of institute was vaccine production. Other contributions in microbiology. Alexander Fleming, he discovered an antibacterial substance penicillin from a fungus Penicillium notatum. While observing a contaminated culture plate of Staphylococcus, he noticed that there was a clear area around the contaminant mold where there was no bacterial growth. Fleming identified it as Penicillium notatum and he named the inhibitory substance produced by the mold as penicillin, but he could not purify this active ingredient. Later, Ernst Chases and Howard Florey developed a method for the purification of this compound. Fleming discovered the hydrolytic enzyme lysozyme, which can break a peptidoglycan layer of bacterial cell wall. Vinogradsky and Bajering. The role of microorganisms in the natural environment was first observed by Vinogradsky and Bajering. Vinogradsky is considered as father of soil microbiology. He discovered that soil bacteria could use iron, sulfur and ammonia to obtain energy, and that many bacteria could incorporate carbon dioxide into organic matter much like photosynthetic organisms do. Vinogradsky also isolated anaerobic nitrogen-fixing soil bacteria and studied the decomposition of cellulose. Bajering isolated the aerobic nitrogen-fixing bacterium Azotobacter and rotenodule bacterium that is capable of fixing nitrogen, later named it as Rhizobium. Selman Waxman, he discovered the antibiotic streptomycin in 1973, which was a major break in the treatment of tuberculosis. Paul Ehrlich is known as father of chemotherapy. He demonstrated that a chemical agent with a selective toxicity that would kill pathogen but not the host cell can be used to treat the disease. Joseph Lister, he is known as father of antiseptic surgery. In 1867, he employed phenol as an antiseptic and disinfectant. He used phenol to reduce risk of infection during surgery. The development of techniques such as electron microscopy, ultracentrifugation and radioactive tracer methods made it possible to probe the physical and chemical nature of bacteria and viruses. Thank you so much for watching my video. Stay subscribed for the next video.